In the well-fed state, there's plenty of glucose in the bloodstream. There's plenty of amino acids. There's plenty of lipids in the form of tags or chylomicrons, packaged in chylomicrons. Okay? So all nutrients are available. However, the preferred nutrient under this condition is going to be glucose. All the actions in the well-fed state are going to be controlled by insulin. Okay? That's an important thing. So start with that. And that means insulin will stimulate the uptake of glucose in muscle and in adipocytes because they have that insulin-sensitive GLUT4 transporter. Okay? That's key thing. So I'll stick with these guys. So glucose picked up in muscle, it's used to make glycogen. It's used for glycolysis. Similarly, amino acids picked up by muscle primarily used to make muscle proteins and enzymes and receptors and contractile proteins, structural proteins. So very important during the well-fed state. In adipocytes, you have uptake of glucose, increase in glycolysis, increase in the intermediates of glycolysis, including DHAP. DHAP can be converted to glycerophosphate. Okay, and glycerophosphate can be used to esterify fatty acids to form tags. Those fatty acids are coming from chylomicrons in circulation. What is the lipid? What is the lipase that does that here? Lipoprotein lipase. That's the one that chews on chylomicrons, picking the fatty acids from the tags. But because the adipocyte has a lot of glucose, it won't use those fatty acids at that time. It will store them in the form of tags. So you have increased tag synthesis, decreased tag lipolysis, because insulin inhibits hormone-sensitive lipase, the lipase that's inside tags, inside adipocytes. Okay, the liver is picking up glucose because glucose is high, not because of insulin, because glucose is high. It has a different transporter. This transporter is GLUT2. The liver is also picking up amino acids, also picking up chylomicron remnants. The use of glucose in the liver is, again, glycogen synthesis, glycolysis. Use of amino acids is really to make other liver proteins, and any excess amino acids will be used as energy. Their carbon skeletons will be used as energy. So the brain is usually pretty happy around after a meal because there's plenty of glucose, it's plentiful, it can pick up as much as it needs and continue its normal metabolism. So I want you to really keep track of the big picture, but as well as important effects of insulin under these conditions, okay? So, and what pathways are going to be activated. For example, glycolysis will be activated, gluconeogenesis will be inhibited. What are the key enzymes in glycolysis that will be activated? Okay. The fasting cycle is really just as important and perhaps in some ways more important because things are less available. Nutrients are less available. So the body has to be a lot more creative in what it does for its metabolic needs. Okay? So the fasting cycle is referred to also as post-absorptive. Okay, again, terminology is very important. Post-absorptive is synonymous to fasting. Obviously, the main thing is you're not going to have glucose. Glucose is dropping. Tags are dropping. Amino acids are dropping. There's less of all three in the bloodstream. So what does the body have to do to compensate for the lack of nutrients? It will go to what is stored. What is stored? Glycogen. It breaks down glycogen to provide glucose. What else is stored? Tags. It'll break down tags, provide fatty acids as an alternative fuel. It will turn over, break down some protein to get amino acids so you can conduct gluconeogenesis using the carbon skeletons of these amino acids. So you are primarily in a catabolic phase where you're breaking large molecules into smaller molecules that the body needs to operate. Okay? So we'll cover some of the key effects on each of the main organs we've been talking about, and we start with the liver. So the liver is the main, although you have glycogen in muscle, the important glycogen storage resides in the liver, because only the liver can produce glucose. The muscle cannot. The muscle produces glucose 1-phosphate, then glucose 6-phosphate. That does not leave the muscle, is unable to leave the muscle. So only liver glycogen is relevant. So glycogenolysis will be increased with the purpose of delivering glucose to the bloodstream so the organs that need it can use it. The liver is the only organ with, the other organ that can do that to a smaller extent is the kidney, that can do gluconeogenesis or has gluconeogenesis. So there's increased gluconeogenesis. So what you think here for in terms of fasting, you are providing glucose to the bloodstream to maintain it at the normal levels. Okay? So the liver's function in fasting is becoming a factory of glucose. It's a glucose producing organ. It's switched from a glucose consuming organ in the well-fed state to a glucose producing organ in the fasting state. In many ways, the liver is kind of altruistic, meaning that it cares more about the rest of the organs than about itself. It is providing glucose for the use of the brain, red blood cells, and other tissues that are entirely dependent on glucose. The liver itself will switch for its energy needs to beta oxidation of fatty acids. It's no longer using glucose in glycolysis. It's using fatty acid beta oxidation for its energy needs. You produce a lot of acetyl-CoA. Acetyl-CoA goes the TCA cycle, produces more energy for the liver. At some point, the liver has enough energy, then acetyl-CoA starts to accumulate. And then that acetyl-CoA is used to make ketone bodies. So the liver then is providing another source besides the glucose, is providing another energy molecules in the form of ketone bodies. Again, The liver itself cannot use ketone bodies, but it's providing ketone bodies so other tissues that can use it will pick it up. And many tissues can use ketone bodies. Skeletal muscle, heart, even the brain can use ketone bodies. That's the only other form besides glucose that the brain can use. Okay? Adipose tissue, key thing in adipose tissue, you always want to think of it as it is our main store of tags, of fuel, lipid fuel in the body. Don't think of adipose tissue as a bad thing. It's really a good thing. We depend on adipose tissue. We don't want too much of it, but we depend on it for our energy needs. When we are fasting, when we are sleeping, when we haven't had a meal for several hours. And just kind of while I am thinking about it, fasting is typically 4 to 5 hours after a meal and beyond. Okay? After 4 or 5 hours from the time you've had your last meal, that's considered fasting. What's also considered fasting is obviously sleeping. If you're sleeping, you're fasting. So you're going to need glucose even when you sleep. So you're going to have glycogenolysis and gluconeogenesis during your sleep. Okay, back to adipose tissue. The adipose tissue is the major depot, if you want, of tags. During fasting, you want to think of two hormones that are going to be elevated in the bloodstream, glucagon and epinephrine. And both of them are going to result in, or going to control much of metabolism. Epinephrine in particular will activate the beta-adrenergic receptor, activating cyclic AMP, activating PKA. PKA phosphorylates hormone-sensitive lipase, which breaks down tags in adipose tissue, producing 3 fatty acids and glycerol. All 4 molecules are released in the bloodstream, transported in the bloodstream. Fatty acids usually are bound to serum albumin, and they are picked up by various tissues that can use them. Muscle, liver, heart can use fatty acids as well. So they're picked up to be used in beta oxidation. To the muscle, remember here, you have no insulin. This is a kind of key point. You have no insulin when you're fasting. You have glucagon and or epinephrine. So the muscle uptake of glucose decreases. It's no longer picking up glucose because of the absence of insulin. Instead, the muscle will start to pick up fatty acids from circulation, use those fatty acids for its own energy needs. Okay? It can use ketone bodies as well, produced by the liver. So it switches to fatty acids and ketone bodies for its own energy. The muscle has another important role in fasting, in that it can provide amino acids. The muscle is rich in protein. Muscle proteins, structural, functional proteins that turn over normally. Muscle proteins typically have a high turnover rate, meaning they're degraded usually pretty frequently and replaced. Degraded and replaced. When you degrade those proteins, you produce amino acids. Those amino acids are released in the bloodstream, transported to the liver, where they can be used in gluconeogenesis. Okay? The brain is really kind of dependent on the liver for its glucose, because the liver is producing the glucose. as well as ketone bodies. Both of them are produced by the liver in fasting. However, ketone bodies in the brain are only picked up after a prolonged fast, not in the first, say, few hours or days. It may take several days, sometimes a week or two, before the ketone body levels reach the concentration that the brain needs to pick them up. Okay? But nevertheless, the brain is able to use ketone bodies. The brain cannot use fatty acids at all because of the blood-brain barrier. Most fatty acids will not cross the blood-brain barrier, whereas ketone bodies can and glucose can.